Hi everyone, today I'm going to be talking about the Natural Products Discovery Course at UMich. So you may be wondering, what exactly are natural products? Usually when people think of the word natural products, they tend to think about 100% organic honey or organic fruits and vegetables, but this is not the case. Natural products are a chemical compound produced by a living organism that can be found in all kinds of forms of nature. What's really cool about natural products is that they can be used to kill all kinds of bacteria, which is very useful when creating medicine. But how exactly do we make something from a plant to medicine? First, we have to look for natural products. We're looking for places where there's a lot of different kinds of bacteria fighting. This could be in certain trees, bodies of water with a lot of organisms like ducks, these funky mushrooms, an anthill, or sometimes even a dead organism. When we find the location that we want, we can then take soil samples from each of the places so that we can grow the bacteria inside of them. We can't easily see bacteria, meaning we have to bring it out of the sample that we collected and grow it. To do this, we have to complete a series of dilutions, which is the process of decreasing the concentration of a solution by mixing or adding water in. After doing this for about 5 times, we're ready to take the water and put it into the agar plate, which will grow the bacteria. After this, we can watch all of our bacteria grow into colorful shapes and sizes. However, there's a lot more work to do to identify what this bacteria is. We have to complete a process of fermentation, which is breaking down the bacteria. Next, we have to find out the name of the bacteria, and if we already know it. This isn't that simple, because bacteria does not come with an identification card or a tag. Instead, we have to look at the genetic makeup of the bacteria. This is also known as the DNA, which carries all the hereditary traits of the bacteria. We can find out the DNA by doing a process of genome sequencing. This could tell us what the name is. However, if we can't identify this molecule or bacteria, we have to actually figure it out. We can use the magical tool of mass spectrometry, which can be useful to measure the mass and charge ratio of a molecule present. This means we can find all the different kinds of molecules inside of this bacteria. We can also separate the molecules inside of the bacteria through another process called chromatography. Chromatography can be used to show the different types of molecules based on how fast they move. But so what we know the mass of a molecule, we still need to find out the structure. This can be done by a very powerful tool called nuclear magnetic resonance, or just NMR. NMR finds the nuclear spins of a molecule using a big magnet, and based on the spins, we can figure out how a molecule is organized. Doing all these steps, we can take this molecule and put it into medicine that's useful for helping people all over the world. Thank you so much to the Astronaut program at UMich and to the Science Communication team.